Thank you, Reha. And I'd like to thank the organizing committee to select me for this talk. I'm from Hong Kong, the University of Hong Kong, from Department of Biochemistry and Center for Genomic Sciences. So as we know that the sequence cost is going down at the Moore's law predicted that it will go down exponentially. The actual cost is actually going down even quicker than the Moore's law predicted. So now sequencing is cheap, but what is next? So my way to think about it is that we need a more efficient way to deposit, store, and process the next generation sequence data. I will touch a little bit about that and then introduce a fast and accurate tool for SNP detection. The other is that after we found the genome-wide association study, found thousands of loci. But how to interpret those loci and how to link those genetic suspected loci to disease and what's the molecular mechanism. So I think that's the next step we should go. So function annotation, as a bioinformatician, we should focus on that. And my group mainly focus on the regulatory regions, such as transcription factor, binding size, microRNA, and non-coding RNAs. And also recently there is a encoded data as published, and we also use this chromatin interaction and histomodification data to help us to prioritize our SNPs. So first I will talk about the algorithm to detect SNPs. So basically we use this FaSD score to measure the probability of a loci to be SNP from the next generation sequence data. There are two parts of this algorithm. One is the alternative score, which is unique to our algorithm. The other is the transversion probability, which is also widely used by other algorithms. So the alternative score we use is that the alternative models, actually, if there is a loci, it could have three genotypes. One is the homozygote, the same as the reference, and then the heterozygote, and then the homozygote different from the reference, right? So if you observe the sequence, this is the depth of the sequence, and this is the number that the allele the same as the reference allele. So if it's homozygote, the same as the reference, then you have 99% of the chance to be the same as the reference allele, but only have a 0.1% chance. We give it 0.1% of chance as an error rate. Then we use this binomial distribution model to feed the reads data at that loci to the binomial model and get a score. So similarly, if we assume is a heterozygote, then you have a half chance to be 50% chance to be the reference allele and 50% chance to be the other allele. And this is also, if it's homozygote different from the reference, then it's on the reverse. Okay, so we feed data on all these three models, and we pick up the maximum. Whichever is the maximum, will assign a score. So if it's a homozygote to the same to the reference, then it's 0 plus the pseudo score. The pseudo score is prevented to multiplication problem. So if it's a heterozygote, then it's 1, and then homozygote different from the reference, the score will be 2. So okay, so we use this TCGA data because the TCGA samples, they do both Illumina array and the FE array on the same samples, and also do the next generation sequencing, so that we can use Illumina and FE as a benchmark. And then we also evaluate, this is our program, and also some other program like MAQ, SNP, GATK, and the BCF tools. You can see that the two array-based platform, the concordance rate are close to 1, so they agree with each other very well. So, but you look at the, for example, the software calling. So this SoapSnip and MAQ both are developed by BGI. So you can see they also agree pretty well. And these two programs, GATK and BCF tools, I think both derived from MIT. They also agree each other well. But when we compare those software with our benchmarks, you can see that our program, on average, did the best. The second one is the BCF tools, and also the other program are not so well. GATK is kind of in the middle. Okay. So that's evaluation based on the array SNPs. Array SNPs usually is more frequent SNPs, text SNPs. But when you apply the algorithm to the whole genome scale, it's different because the whole genome scale, most of the core will be homozygous to the reference, which is AA allele genotype. So in order to remove this AA, since 99% of the core are AA, so if you use the concordance rate to measure, then you won't distinguish the difference between the programs. So therefore, we use this non-reference concordance rate to measure this on the genome scale evaluation. So the way we measure it is that we use a very high throughput, high depth genome sequence, which is more than 30x genome sequence. And then we use the MAQ to call SNPs and use that calling as result as a golden standard benchmark. And then we randomly sample the reads, 10% of the reads, from that high depth genome and become a low depth genome, and then we use other program to call it. The calling result, because at the low depths, the reliability will be lower, and then compare with the high depths, which call from the MAQ, and then evaluate the long concordance rate. Overall, you can see that, of course, the concordance rate dropped compared to the previous slide, but still, relatively, our program performed very well, both on chromosome 21 and 22, because these two chromosomes have different GC contents. That's why we select this one. So you look at the SNPs called by various programs, the overlap part. You can see that if the SNP is called by all programs and either GATK or BCF tools, you can achieve pretty good accuracy. This is the number of SNP called, and this is the accuracy. So 99.7% you can achieve. But if you only call by an individual program, for example, our program, we can achieve a 63% accuracy. But GATK, if only called by GATK, is 43%, and the BCF2 is 36%. Okay. So we also evaluated the running speed based on the tenfold human genome, both on the server, which is single CPU used, and the PC. You can see that it will only take half the time to finish the job compared to GATK, and also much more efficient than BCF tools. Okay, so that's the program. I briefly introduced the program, but I advise you to go to the publication and look at it in detail. We also did lots of other evaluations. Okay, so the other I want to mention is the genome-wide association study. We can see that so far over 1,500 genome-wide association study has been conducted, and over this, I think thousands of loci were discovered to be associated with disease, category by the NHGRI website. So we are thinking, what can we do as a bioinformatician? So what we did is that we created this GWASdb database. So we did extensive function annotation on those genetic variants. For example, you know the SNP summary, the allele frequency from 1,000 genome project and from the HapMap, LD plus, and also how does the SNP map to the gene region based on this ref gene annotation, and also microRNA, whether it's a transcription factor binding site, and also the regulatory effects, especially focus, transcription factor binding site affinity, splicing, and also microRNA target prediction. And also this, if it's a protein coding region, then how it will affect the protein's function, the cross-species conservation, and also expression the EQTL information and disease association. Furthermore, we also map those genetic variants to the disease ontology light and also the human phenotype ontology. And also we incorporate pathway protein-protein interaction information put into this GWASdb. We do one thing further is that instead of just get the SNPs, which is genome-wide significant SNPs from the NHGRI database, we also go to the literature paper by paper to dig out those that are not at a genome-wide significant level down to the 10 to the -3 p-value and get the other SNPs that may be useful for the users. So we got actually 20 times more than the GWAS catalog in this database. Now we actually plan to on real time to curate from the journal papers. So we probably will be more real time than the GWAS catalog in the future. So once you have the SNP, you can input the SNP ID, and then this is the browser we developed. This is the SNP you are interested in, and also the many function annotation like gene region, DGV, stuff like that will be here. And also many regulatory annotation will be on the right panel. So we, as we mentioned that we also map it to the disease ontology. This is the alphabetical order of the disease summarized, and you click on each disease, this Manhattan plot will show all the genetic variants that's so far in our database. And you click any of these variants, it will give you the item. Either you can go to the publication, original publication, or go back to the GW browser, which I showed in the previous slide. So that's the database. And then we also did some prioritization on the SNPs. The reason the need for prioritization is that the array SNPs usually get from the tag SNPs. But tag SNPs are only 5 to 10% of the total SNP. For example, the DB SNP now have over 50 million records in the database. But the most recent Affymatrix array only have around 0.91 million SNPs. So you only select like 2% out of this. Then if even your chip find some SNP that are significantly associated, but the GWAS SNP doesn't mean that it is really a functional SNP, right? So because GWAS tagging is based on the LD information, even so they are in perfect LD, but the function annotation are different. So with the coming of this encoded data, you should use those, for example, DNA lace hypersensitive sites and the chip seeking binding sites, those data to reprioritize those SNPs and give it the function meaning. So if you do function validation only on the GWAS tag SNP, then most likely you will fail find the function. So you got to find the real functional SNP. So that's the rationale. The way we also do it from our GWAS database, we categorize into different categories and then calculate the enrichment scores, and then use this enrichment score as prior to prioritize those SNPs. So this is the pipeline we use to prioritize those SNPs. So the genomic location, of course, it matters, and the conservation, folding, transcription factor regulation, splicing, microRNA, protein coding, and also EQTL, post-translational modification. All these we put together and then get the final functional score. So we actually evaluated this pipeline, see whether it works. Basically, we randomly select 100 SNP from the DB SNP. You can see that through our pipeline, reprioritize scheme. Only one shows significant high score. This is the positive data set, which is from Almin data set. You can see that a majority of the SNP show pretty high score. And this is the GWAS catalog 100 SNPs. You can see that it also pretty reasonably well scored, but many of them are just like random. And once we use LD to reprioritize the other algorithm, we can see that many of these score very low and improve the scores. You can see that this prioritized scheme with LD really works. Okay. So we further not only annotated those in the GWASdb database, we also annotated this in the whole genome base. So right now people can use it. If you have any RSID, the DB SNP ID, you can go there and we can immediately give you function annotation. Even you don't have a DB SNP ID, if you input the chromosome location, we can also give you function annotation. So on this web server. So the other drawback in the function annotation is these long-range interactions. You can see that many of the enhancers, especially, they are several hundred kb away from the target gene. So traditional annotation is that you just use a hard cutoff, for example, 10,000 upstream of the transcription start site, and you define this as a regulatory region. But the thing is that sometimes enhancers are far away from those regions. How do you link this enhancer to the target gene? That's a problem. So as the encoded data come out, so we have the opportunity to use those data and to annotate, to relink the GWAS significant SNP with gene relationship. So I will briefly introduce a little bit on the 3C, 4C. Basically now they have high throughput and low throughput technology to map those enhancer and promote relationship. For example, the 3C technology, they can map one-to-one relationship, and the high C, they can map all-to-all relationship. Basically, they can link the enhancer to the promoter. So we take advantage of their data. So if user give a list of SNPs, we can find those chromatin interaction information. Also, we have this prioritized pipeline based on the this active marks information in insulator. Also incorporate CHIA-PAD, 5C, and high C data, and also the population-wise the conservation information. Okay. So put together, we can reprioritize. This is the example that this is the interface of the our result. Once you give the list of the SNP, so we can give you those, and also function annotation. Here's the example. Is we use this LDLC example. We have the publication give us 17 associated SNP in this region. And after our pipeline, we found 5 significant regulatory variants. So we signal especially the leading one is this guy, RS, this guy, and is on the top of the table, and also the significant score change from 10 to the minus 42 to minus 46. And also we found 2 active enhancer markers, H3K27 and DHS on that side. And also we found the binding side, which the 2 allele have different binding affinity around this binding side. And the conservation, when you use this HEP G2 cell line to annotate it. But interestingly, when you use other cell line to annotate it, you don't find this signal. So that shows you that the tissue-specific annotation, if you use these epigenetics and chromatin interaction marks. Okay. So in summary, I introduce a SNP detection program which is fast and accurate. I also introduce a GWASdb database for genetic variants from the genome-wide association study. We have this genetic variance prioritization tool. You can use it at a whole genome level and doing extensive function annotation. And also we developed this GWAS 3D database, which can function annotation of the regulatory variants from chromatin interaction and epigenetics data. So I want to stress that all the tools are available freely at this website. You're welcome to go to use those tools. So finally, I want to thank my lab member, Moonin Lee is the guy that my student did the GWASdb, GWASrep, and GWAS 3D, the major developer, with the help of Pan Wen, Feng Xu, and Wei Xin did the FaSD algorithm program, and I needed help on the GWAS 3D database. I also want to thank my collaborator, Professor Pak Chen, Maria Wong, Junwen Xia from Hong Kong U, and also Stephen Chanlock from NCI USA, and also the funding from RGC, HMRF from Hong Kong, and the NSFC of China. And also thank you for your attention.
You got time for questions? Yeah, there's one already.
So actually, when you combine all types of so many types of different information to the presentation, it looks pretty cool. So my concern is what is the major methodology which we evaluate those SNPs and to get a new score for that.
So that's a very good question. So we are very careful when we develop this pipeline. Basically, we started with biological rationales. And then once the pipeline is finished, then as I show you that you have a random selected SNP number, and you have the SNP that are known, experimentally known to be functional, like the SNP annotated in the ORMIN database. And then you score those SNPs, right? You look at the distribution. If the distribution are totally separatable, then your pipeline works. Otherwise, it doesn't work.
Did you compare these results with Reprioritizing by Stanford University? They also have some communication about the function based on the GWAS and letting the encode.
We didn't. Yes.
Another question, yeah?
So first of all, thanks for putting up this nice database. Thanks for the is it also available as a full download, GWASdb?
Once you specify a disease or the annotation, I think we have a button you can click and download the data. for a specific disease. But for the whole database, it's too big, so